Today is going to be the first of a two-part lesson, first part, and it is going to be DNA technology. Whoop. All right, well, on the last episode of CSI Tampa, a young woman was found murdered in an alley behind Daily Eats, and there were no traces of the suspected murderer. There was no hair, no fingerprints. However, forensic investigators were able to detect 10 cells underneath the victim's fingernails. These cells may provide a DNA sample of the potential murderer. However, 10 cells is pretty pathetic. All those cells do have a nucleus, and those nuclei have a full copy of the suspect's DNA, but that's not a lot. You need way more. So we have to make a lot more DNA to try to get to the bottom of this murder investigation for CSI Tampa. So we are going to do one of the DNA technological, what's the word I'm thinking of? DNA technological procedures that a lot of forensic detectives and in Tampa Police Department or Hillsborough Sheriffs, Florida Highway Patrol, whatever, and due to trying to uncover the owner of DNA, and that would be called a polymerase chain reaction. Now, this method is all about producing millions and perhaps billions of copies of DNA when you originally had very little to start with. So next week you guys have a test, and I have to type up your test. And when I print out your test, when I type up your test, I have to print it. I don't want to use up all my ink toner to print out 30 copies of the test. So when I print out one copy, where can I go to make more? What do I do? What machine do I go to? A copier, right. So in the PCR procedure, you need a copy machine. This copy machine is called a thermocycler. When you hear the prefix thermo, what is that referring to? Heat. So this is a machine that regulates the temperature for the PCR process. In this PCR process, you need a few, you have a few needs that are necessary to actually conduct it. The first thing that you need is you need your DNA sample. Those little cells that were found underneath that woman's fingernails. Next would be some free nucleotides. You need to be able to have the ingredients to make more DNA, and the ingredients to making DNA are nucleotides. And then you need some enzymes, like the ones that you use in semi-conservative replication. But there's some good news here. You're not going to need a whole lot. All right, and this procedure is actually pretty simple. It's 3 stages, and in 24 hours, you could, actually less than 24 hours, you could get billions of copies of DNA when you initially only started out with, in this case, 10. All right, so this is a 3-step procedure. Here is step 1. The sample is heated to break the hydrogen bonds. Now, on the first day of molecular genetics, when we, let's see, that was about 3 weeks ago, we spent the first day talking about the basic structure of DNA, and I told you that there are hydrogen bonds that connect 2 parts of DNA. Can any of you think back to what part of a DNA molecule is held together by hydrogen bonds? Think about that. Jasmine? The bases. Do you remember how many hydrogen bonds connect A to T? 2. Anybody remember how many hydrogen bonds hold C to G? 3. That's right. Now, guys, these are hydrogen bonds, or H bonds for short. Now, the next part you don't necessarily have to draw, although I will say that when I was looking over your semester reviews, some of you got this wrong, so now is the time to learn from your mistake. On one of your questions on the first unit of your semester exam review, you were asked to draw 3 water molecules, label the positive and negative sides, and draw hydrogen bonds connecting them. So let's say here's a water molecule here, here, and here. Well, hydrogen bonds hold them together from one hydrogen to a neighboring oxygen. These hydrogen bonds can absorb a tremendous amount of heat. However, everything in nature has its limits. And do you know at what temperature will these bonds break, causing liquid water to become a gas? At what temperature in Celsius? 100 degrees Celsius, which is the temperature in which water boils. What happens at 100 degrees Celsius is these bonds just get so outmatched by the heat that they break. And now there's nothing holding the molecules to each other, so they're free to float away as a gas. Well, the same logic holds true with this. When you heat up DNA to about 100 degrees Celsius, these hydrogen bonds will break, which will separate the 2 sides of the DNA, which is what you want to happen. Keep in mind, human body temperature is about 38 degrees Celsius. You're going to heat this up all the way to 100. So that is going to pose a problem, which we'll get to momentarily. All right, so the DNA is basically done this. Here's the before, here's after. You have successfully separated the 2 halves. That's what you want to do. Step 2. The sample is cooled enough for base pairing to occur with free nucleotides. The sample is cooled enough for base pairing to occur with free nucleotides. You're going to cool it down to about 70 degrees Celsius, which is still nearly double what the human body temperature is. So that's still really, really hot. Temperatures that the human body should never have exposure to, which obviously your DNA is going to have some trouble getting that hot. Now, step 3. This is where it gets interesting. Guys, it is far too hot for our polymerase, which, as you all know, DNA polymerase will make a complementary side of DNA based on the template. Well, here's the issue. We have a problem. DNA, and I should say human DNA polymerase, cannot survive the high temperatures. Now, an enzyme is a protein. What is that D word when proteins get too hot or too acidic and they start to break down or stop? It's called denature. Our polymerase will definitely denature when it gets that hot. So here's the cool thing about life on this planet. No matter if you are a mammal, bird, reptile, amphibian, crustacean, arachnid, insect, worm, or single-cell prokaryotic bacteria. We all have the same four bases, A, T, C, G. Everything on this earth is A, T, C, G. Even viruses, which technically aren't even alive, they are A, C, T, G. So there's an organism out there that can tolerate these high temperatures. They're found in one of the most they're found in one of the hottest places on Earth. And we have found them, and we are going to use its DNA polymerase to bring the DNA together in these hot conditions. Can you tell me where could we find one of these organisms that lives in one of the most hottest places on Earth? Andrew? You got it. A hydrothermal vent is so hot it will literally make the meat on your bones ooze off. That is for you. But for these little single-celled bacteria, it's heaven. In fact, just a couple weeks ago, scientists found 30 new species of critters that are living inside a hydrothermal vent, which is basically an underwater volcano. Well, this little creature is called Thermus aquaticus. Hot water is basically what that name means. It lives in hot water. So we're going to call it TAC for short, T-A-Q, like Thermus aqueous, AQ, or aquaticus. We are going to use TAC polymerase to make more DNA. And we're going to use it to bond the complementary nucleotides to the template nucleotides. It will not denature at these high temps. I mean, it loves these high temps. This organism lives practically in an underwater volcano. So its enzymes, its cell wall, everything is able to withstand these high temperatures. And so you do this process, and you went from having one copy of DNA to now you have two copies. And then 2 becomes 4, 4 becomes 8, 8 becomes 16, 16 becomes 32. It just goes on and on and on. And within just a few hours, you can have billions of copies. Plenty to work with when trying to do some forensics to figure out who killed this woman. So I'm going to show you a video in just a moment. It's very short, and it kind of just shows how simple this process is. But now that you have, let's say, millions or billions of copies of DNA, what is done with the DNA now? So after you have billions and billions and billions of copies, now what? So before we get to that, I am going to switch over to YouTube real quick. If you're watching at home, I'm going to have to pause the video because I can't show a YouTube video on Zoom. But if you go to YouTube and you type in polymerase chain reaction, I want you to find the video that is 1 minute and 28 seconds. It looks like this. This is what I'm going to focus on. So right now I'm going to just pause the video and we'll come back in just a sec. So now that you have this DNA, what do we do with it? That's going to get us to the next part. This is called DNA fingerprinting. When you fingerprint someone, oops, you are matching their, something that's very unique to that person, Unless you're even if you're an identical twin, you have your own fingerprints. So in order to do some DNA fingerprinting, and the main process I have for you momentarily, we're going to need something called a restriction enzyme. On Tuesday, you guys are going to do a simulated biotechnology lab. And one of the questions on your post lab is going to ask you, do restriction enzymes occur in nature? Well, here's the answer to that question right now. They are present in prokaryotes. What are they used for? They defend against viruses. Now, how would they defend against viruses? So let's say I have a sample of bacterial DNA. And just like any other organism, bacteria can be infected by a virus. The virus that gets a bacteria is called a bacteriophage. It's a virus that targets bacteria. And let's say this highlighted portion of this bacteria's DNA just got infected by a virus. Viruses are called obligate parasites. They have to have a host to survive, and they need the host DNA to make more of the viral DNA. Well, bacteria doesn't want its DNA damaged by bacteria, so what a restriction enzyme does is delete that infected DNA. And that new DNA will come back. So basically, it's like if you have a part of your body that is infected, you cut off that body part. That's kind of like what the bacteria do. Except for them, the DNA will come back. Your body part won't. So a restriction enzyme, they target specific gene sequences. And they cut the DNA at that targeted sequence. So thanks to the PCR reaction, we now have two sequences of DNA from this person whose cells were left underneath the victim's fingernails. Sequence number 1 is going to look like this. GATA CAG GCAT. Sequence number 2 is going to be GATTGGACGGT. Now, what if I told you that the restriction enzyme that we are using here, this particular restriction enzyme, cuts between two guanines that are side by side? If I told you that you have a restriction enzyme that will cut two guanines and will separate those sequences, you would have to look at the sequences and find where that's going to happen. So it's going to happen here, here, and here. So we're going to have 1, 2, 3, 4, 5 different segments of DNA. So we have 7 bases here, 4, 5, 4, and 2. 7, 4, 5, 4, 2. We will now do a procedure called gel electrophoresis. DNA, the name is a lot scarier than what it actually is. DNA gel electrophoresis. If you go to college, you'll very likely do this in one of your labs. It's pretty simple. The way that this procedure works is that it uses electrical current to separate fragments of DNA by the size of the fragment. Now, it's something to consider. DNA is negatively charged. It has a lot of negative ions, which are anions. That's going to play a role here. You all should know that anything that's negatively charged is attracted to something that is positively charged. So what we're going to have first is we're going to have something called the gel agar. It's like kind of like a brick. It's shaped like a brick. It's probably about the size of your hand, and it's made of a gel called agar, A-J or A-G-A-R, and it can have a gel in it. Well, over here on the left, we're going to have the negative electrode. The DNA is going to be repelled by this side, and on the right side, we're going to have the positive electrode. The DNA will be attracted to that side. Now, the way that this works is you're going to have the smallest DNA fragments move their way across most of the gel agar. And you're going to have the larger fragments move hardly any at all. So I have a kind of an analogy for you. Reagan has a piggy bank at home where she's been saving up coins since she was a little girl, and she has a couple $1,000s in coins now. So she's going to just smash it and get to all that money. Well, this piggy bank has dimes, nickels, pennies, quarters, and even half dollars, which are pretty big. Which coin is the smallest in size? Not value, but size. A dime. So we would expect the dimes to fly away the furthest. The half dollars are pretty big. They're like that big, double the size of a quarter. So they're probably hardly going to fly anywhere. So the same logic goes here. The smallest DNA is going to move across this brick or this gel agar, and the larger DNA is hardly going to go anywhere at all. So what we have here are wells. Wells are where we actually insert the DNA samples, all of it. Now, this first well is going to be the crime scene DNA. Remember that this is not the victim's DNA. This is the DNA that the suspect left behind. So this is going to be our crime scene DNA. And investigators were able to find 3 persons of interest. They're not arrested. They're not accused of anything yet. They're persons of interest. And we were able to obtain a warrant from a judge to get a genetic sample from these people. So we're going to have suspect number 1, suspect number 2, and suspect number 3. And due to DNA technology, there have been a lot of people that have been either found guilty of a crime that they were initially deemed to be innocent of, and the opposite. There's been people that have been found innocent of crimes that they were accused of doing. There's a guy in Tampa that was thrown in jail in the 80s, and he just got let out, I think, in November. And imagine it's like going in time. He was stuck in jail for more than 30 years, and he comes out, and he's like, whoa, wait, we have a hockey team? Because back in the 80s, the only team around here was the Bucks. Lightning came in 92. Rays came in 98. He's like, wait, we have a hockey team? And they won the championship twice? We have a baseball team? Like the Bucks changed their logo? Like Tampa is a big city now? Like a lot of stuff. So this guy just went through time. What the heck is an iPhone? He had no idea. And DNA technology proved his innocence. So this is the kind of thing they do. So we put the DNA into the well, and this is what we get. We get some DNA patterns. Here are the heavier fragments. Then we have some lighter ones. And then we have some very light ones here. So this is the pattern. This is the DNA of the crime scene. Now let's compare it with suspect number 1. Okay. Suspect number 2. And suspect number 3. Let's compare. This is what we're looking at. Here, here, there, there, there. All right, suspect number 1. Looks like we have 1, 2, 3, 4, 5, 6 parts that match up. But we have this and this that don't match up. All right, next one. Suspect number 2. Both of these match up. Both of these match up. And these small sequences match as well. But this is out here in the middle of nowhere. The initial crime scene did not have that segment. And then we have suspect number 3. So based on this evidence, guys, which suspect is the leading candidate for being the murderer? Suspect number 3 is the best match. It's like an identical match. But the way the criminal justice system works is you can't just say, he did it, we got him. You have to test over and over and over. Other people have to do the test. You know, that's what science is all about. You come to a conclusion. Other people should be able to come to the same conclusion you did using the same method that you did if your results are to be considered legitimate. So if I were to do this, then Reagan should be able to do that, get the same results. Dan should be able to do it, get the same results. And not only do we have different people doing the same procedure, here's something else that we use. We're going to use other restriction enzymes. So you're going to use various restriction enzymes to repeat this procedure. In science, the number one thing that scientists are looking for are mistakes, because sometimes a conclusion can be too good to be true. So they want to look for any flaws in the design that might have corrupted your outcome. And that's why it's important to get other scientists to repeat your work. That's what we call like a peer review, so that they can see, oh, you didn't do this right, or oh, that's not correct. You did something wrong. You can't just do one test and say, oh, they're done. Throw them in jail. I got the murder. You have to do it over and over and over and over again. Have any of you ever seen the film Straight Outta Compton? Chronicles 4 or 5 rappers from the late 80s, early 90s. Well, one of the rappers' names was Eazy-E, and he died of HIV in the early to mid 90s. Well, he got HIV from having a lot of unprotected sex with strange women, and he got HIV, which is an STD. Well, when the doctor came to him in the hospital bed and said, we did a T cell count, and your T cell count is very, very low. You have HIV. And he was just stunned. He's like, that's not my test. You need to take that back and do it again. And the doctor said, we did it again. We did it like several dozen times. And it came back the same each time. See, the doctor didn't just do one test and say, hey, you have HIV. You do it over and over. You double and triple and quadruple and so on and so on. Check your results before you can finally conclude, yep, this is it. You know, if I were to grade Dan's test 20 times and it came back the same every time, that's going to have a lot more confidence in it than grading it one time. All right. So this is what a lot of forensic detectives in both city police departments and the FBI or county sheriffs use to try to find who the perpetrator is. Okay. All right. Let's start to come to a close here. That would have been one day's lesson, but this is a 2-part lesson, so we're gonna get a little bit into tomorrow. And I'm gonna show you guys the final video here. Just want to get like 5 more minutes out of you. What we're gonna transition to next is genomic technology. A genome is an individual or a species' entire genetic code. Back in the 90s, private scientists and government scientists spent 14 years, 12 or 14 years, interpreting the entire human genome from front to back. That's where we learned about introns. We learned that there's these segments of DNA that do have information called exons, and there's these segments that we have no idea what they code for. For all we know is junk. Those are introns. So when you have genome technology, you are actually manipulating the genome. And the first thing I want to bring up is a GMO. Anybody know what that stands for? Ryan. A genetically modified organism. This term is a non-scientific term. But the definition of a GMO is any organism, plant, animal, fungus, bacteria, anything that has had its genome, which is its DNA, modified in some way. There is something in our world today that has become very, very, very prevalent, and it's called a conspiracy theory. Unfortunately, guys, there's a lot of people out there that are either uneducated or they have something called confirmation bias. A confirmation bias is where I could tell Hartley the world is round, the earth is a globe. And I could take her to space and I could fly around the earth and say, Hartley, do you see? Well, the thing with confirmation bias is even if you show someone evidence that proves their stance wrong, they ignore it. They deny it. They don't believe it. And so when you think that you are saying, oh, I've got them this time, no way they can deny this. They do. They ignore anything that rejects their stance, and they only listen to things that confirm what they think. So what I'm getting at is with GMOs, there is zero scientific evidence that says that it's bad for you. There is no evidence that there are health problems. There's no evidence that there's cancer or anything like that. Just kind of like with vaccines. But there are people out there that will think, oh, it's bad, it's bad, it's bad. So I'm not gonna, I don't want to touch it, I don't want to look at it. There's no evidence that says it's bad. And in fact, you guys eat a tremendous amount of GMOs. There's 8 billion people on this planet. Most food is genetically modified. If it wasn't, you would have billions, billion with a B, people die. Like large percentages of this global population die because there would not be enough technology or enough food to feed all those mouths. And then all you would have is like all natural food grown without any modifications. There would be war for whatever food is remaining. You know, here in Florida, we have GMOs everywhere. Citrus is a GMO crop. Sugarcane is a GMO crop. Strawberries are GMO. Anybody ever had the cotton candy tasting grapes at Publix? That's a GMO. There's something called a pine berry. It is a strawberry that tastes like a pineapple. That's a GMO. If you take a look here, look at this cow. That is a ripped cow. Well, that's a lot of meat, which means that's a lot of muscle, which means that's a lot of beef for Burns and Charlie's and Fleming's to have for their customers to eat. The more muscle a cow can have, the more muscle there is to sell, more money. This next one is called a potato and a tomato hybrid. It's called a pomato. It has tomatoes above the ground and it grows potatoes below the ground. So these are all GMOs. And then one more thing for you, which I think I'll have to get to tomorrow because I want to show you a video. I was going to show you some puppies that glow in the dark, but that'll be for tomorrow. So the last thing I want to share with you guys is a video from Jimmy Kimmel, which is one of the late night shows on TV, where he goes around asking people what are GMOs and why don't you want to eat them? So I'm going to stop the sharing now. Let's stop this.